we're going to put them through these diagnostic filters. And then in the end, we want to kind of have this nice, neat package of confirmed examples of rescue. And the three filters that we really focused on was there needed to be some risk of extinction, whether that was from demographic data, from simulations, or from natural history information. There needed to be some evidence of allele frequency change due to selection or gene flow or some other mechanism of evolution. And we were pretty loose. The population needed to persist. So we weren't prescriptive about a population recovery. Instead, we were really focused on this idea of the population avoids extinction. So to kind of walk you through the way that we were thinking about this, I'm just going to go through 3 of our case studies, kind of describe how we decided which ones were evolutionary rescue and which ones were not evolutionary rescue. So first, we have butterflies that live in the Nordic islands, and in response to habitat degradation, started to evolve alleles that helped them disperse. In populations that underwent moderate degrees of habitat degradation, these alleles were able to allow the population to persist. Whereas when habitat degradation was really extreme or when there was complete habitat loss, these alleles were not sufficient to allow population persistence. So we have some populations where evolution could save the population and some populations where extinction happened. Looking to coral systems is another example. Simulations done by Rachel Bay and colleagues show that coral evolution can help prevent extinction so long as climate change is relatively moderate. So again, this kind of repeating theme of moderate environmental change could allow adaptation to keep pace and allow the population to persist. And then our third example is the anole lizards in Texas. So in Texas, they have these really extreme cold snaps that you might have read about in the news recently. And these researchers found that pre and post cold snap, there were clear changes in allelic distributions for these populations. So thinking about these 3 examples, the way that we categorize things was our lizards were an example of evolution without extinction risk. So this wasn't rescue. There wasn't really any clear signal from the demography or from the natural history of an extinction risk in these populations. There was just some sort of microadaptation happening. Whereas as we start to move into our coral example, we have simulations and natural history kind of supporting a narrative of evolutionary rescue. And then what we consider kind of our highest level of evidence is these repeated evolution experiments across a metapopulation, where you have some populations that are persisting, potentially due to evolution, and some populations that are going extinct. So our literature search identified about 480 possible examples of evolutionary rescue. Using our diagnostic criteria and things like looking for genomic or phenotypic data over space or time that are showing contemporary evolution, we narrowed this down to 41 case studies. Of these case studies, we said 13 were predicting evolutionary rescue. So showing that there's some variation that exists and creating a narrative that this variation could potentially rescue the population from extinction, or using simulations to show that evolutionary rescue was a possibility for these populations. We had 19 examples of ongoing or successful evolutionary rescue where evolution was supporting the persistence, recovery, or niche expansion of a population. And then 9 examples of what we called failed evolutionary rescue, which is kind of a grab bag of different things. So this includes things like researchers looked for evolution in a trait and the trait wasn't heritable, or the population continues to go extinct despite evolution. So as we're thinking about these studies, we wanted a way to kind of visualize them and summarize them across some sort of mental map, if you will. And the idea that we had was we thought about, we can imagine each of these case studies as a point, and we can plot it along an axis of where's the state of evidence for evolution? Is evolution predicted or occurring? So along our x-axis, when we're further to the left, these are case studies where there's variation that exists, or there's some sort of prediction of rescue. As we move to the middle, we get some simulations suggesting evolutionary rescue is possible. And on our rightmost side, we see examples where there's some sort of sweep or clear genetic change that has occurred in conjunction with evolutionary rescue. Our other axis is really thinking about the population state. So on the bottom, is the population declining, or potentially has the population even gone extinct? In the middle, we have populations that are persisting, maybe not doing too hot, but they're still there. And then on the top, we have populations that are actually increasing or recovered to historical baselines, maybe expanding their niche. So looking at all of our case studies here, this is the kind of way that we plotted them out along this space, where the point color gives the type of environmental stress that the populations are facing. So I just want to highlight a couple of really fun examples and show the ways that our predicted successful and failed rescue map onto this space. So successful or ongoing evolutionary rescue, we said, was this kind of upper corner. And a nice example of this is the killifish that Andrew Whitehead and his lab studied, where they've evolved evolution to really toxic environments. This allowed kind of niche expansion or an ability to thrive actually in environments that they would normally die in. On the left-hand side, we have our kind of predicted evolutionary rescue. Again, that's similar to like Rachel's coral study. And then along the bottom, we have examples of failed rescue. This includes things like this study by Clark Wolf et al., thinking about how penguins are failing to track climate change due to climate variability, and how that will ultimately lead to extinction of these really adorable penguins. The other thing that we noticed and we wanted to call attention to was these kind of diverse mechanisms of rescue. So obviously, we see selection leading to evolutionary rescue. A nice example of that is insects which become wingless after deforestation, and this allows them to kind of persist in a new niche space. We also see evidence of gene flow and introgression allowing rescue. For example, Darwin's finches responding to extreme drought, where introgression between two species facilitated persistence of the populations. And then finally, we see things like genetic purging also allowing rescue. So what's interesting about this case is instead of sweeping some sort of magical adaptive allele, we're just getting rid of genetic load, and that's allowing us to recover. At the end of our paper, we kind of go into this framework, thinking about how can managers facilitate, promote, or impede evolutionary rescue. So either facilitate rescue that's ongoing, promote or jumpstart rescue, or impede and slow rescue. So we can imagine, if we're a natural resource manager, we start to see our population size declining. We don't know what its future is going to be. What can we do to hopefully have our population recover, maybe using some evolution? So thinking about our adaptive alleles in green there. So the first thing is we can reduce environmental stress. This is a way to buy time for evolutionary rescue. It tends to lead to a much smaller drop in the population size, which also facilitates rescue. Second, we can think about preserving extreme alleles. So for example, in the case of Tasmanian devils responding to devil facial tumor disease, there's some evidence for adaptation in certain populations. So making sure that populations with individuals who are adapted to devil facial tumor disease are persisting is really important to make sure those alleles can continue spreading. And finally, we can even think about things like assisted gene flow, or maybe in some cases, assisted evolution to actually introduce adaptive alleles into populations and directly jumpstart the rescue process. This is something that's in the works for Australian voles responding to invasive cane toads, for frogs responding to BD fungus, and for some coral species. I want to end this vignette by thinking about the kind of future of evolutionary rescue and where I see the field going. So evolutionary rescue is a really powerful way for a population to persist in response to some sort of novel environmental stressor. But at the same time, the future of the population, is the population going to be thriving, let's say, is still up for debate. And I really like this case study focusing on Atlantic salmon as an example of this. So these researchers found that after really extreme reductions in water flow, salmon evolved smaller body sizes to accommodate this change in water flow. And they allude to this idea that this could be evolutionary rescue. And it's certainly a huge conservation win that this population persisted despite really extreme environmental change. But I think a question is, if we start to extend our time scale or our time horizon that we're looking at, what does this mean for the population as it faces novel stressors that continue to come its way? So what happens when this river gets really warm or when this river gets really polluted? Does evolutionary rescue in response to changes in water flow affect a population's ability to respond to other novel changes? The other thing that I want to point out here is that there's some potential trade-offs in terms of management goals and outcomes of evolutionary rescue. So again, small fish persisting in the river is great. We like having salmon around. But at the same time, these small fish migrating to the ocean and experiencing fisheries might actually be misaligned with management goals that want big fish to be caught by our fisheries. And so there's this kind of emergent trade-off that could be happening when we start to look at different management goals that we have and how evolutionary rescue maps onto those goals. So for my second little vignette here, I'm going to talk about a mathematical model that I created that thinks about evolution of parasite resistance in salmon farming contexts. So specifically thinking about how sea lice evolve resistance to chemical treatments. And this work was done in collaboration with folks from across Canada and the Western US. To kind of motivate this a bit, disease control is a really central concern in salmon farming. So when fish have parasites, they're ugly, they're smaller than fish without parasites. We don't want our fish on farms to have parasites. Moreover, when those parasites move from the farm setting into the wild, they can infect wild juvenile salmon, and this can depress population productivity of wild salmon, which leads to negative conservation outcomes. So a perhaps really obvious solution to this problem of not wanting parasites is that we can use really intensive treatment. But this has two downsides. The first is that treatment can be very costly to apply, and it might not lead to necessarily prettier or larger fish on the farm. And second, it also leads to really strong selection for resistance to treatment. And this can erode our kind of management gains over the longer term. Simultaneously, there's some ideas in the literature that gene flow from wild lice can actually slow this selection. And so there's this kind of push and pull between lice on the farm infecting wild fish and depressing their population, and wild fish bringing parasites to the farm and providing kind of what we call an ecosystem service, where they slow negative evolutionary outcomes. So I wanted to explore all these dynamics, in particular thinking about how do different treatment strategies change the number of wild fish that we have? So I call that my conservation outcome. The number of times that we treat and the cost of treatment, which I call my economic outcome. And the degree of resistance that develops on the farm, which I call an evolutionary outcome. So to answer this question, I created a dynamical systems model thinking about three environments. So we have a domestic environment, which is where our fish farm is. We have a link environment right next to our domestic environment, so parasites can move between these environments. And then from our link environment, we have a wild environment. And so wild fish are moving from our link environment to the wild environment throughout their life cycle. So in each of these environments, I model a fish population, and each of those fish populations have some parasites on them. Like I was mentioning before, parasites are able to move between the domestic and link environment. And then in the wild environment, parasites are exchanged between fish. In the domestic environment, we have some sort of treatment going on. In this case, we think about discrete treatment pulses, which is relatively realistic to how salmon farms treat parasites. So what happens is, as the lice burden increases, we reach some predetermined threshold of lice that we find acceptable. And at that point, we treat the lice, which kills off most of them. And then the cycle just keeps repeating. So we kind of have this growth and decay of the parasite population. We can define our treatment by both the threshold by which we choose to treat at, and then the efficacy of the treatment, so how many lice are actually being killed each time that we apply treatment. Finally, I layer evolution on top of this using an invasion analysis, where I have both the susceptible lice to treatment, and then I model a second population of resistant lice, which are our dark orange circles here. And the questions that I want to explore are, how do our different treatment thresholds and effectivities affect our conservation outcome, the wild fish population size, our economic outcome, how many times we're treating, the kind of parasite outcomes associated on the farm, and then our evolutionary outcome, what's our ratio of resistant to susceptible fish at equilibrium after some period of time. So to kind of understand how these different outcomes are trading off with each other, I drew on the economic sufficiency frontier framework. So here, instead of trying to figure out how much a wild salmon was worth in dollar amounts, I just directly compare the number of wild salmon to our different outcomes. So for example, the juvenile salmon population size compared to our economic cost. And on the right, the juvenile salmon population size compared to our proportion of resistant lice. So I want to walk you through what's going to come up on the figures, and then we'll look at the results together. So I'm going to plot a bunch of different simulations on these graphs. The points are different treatment effectivities, and then the lines between the points indicate that those share a treatment threshold. I'm going to plot a bunch of points, and then I'm going to look at the shape that the outermost points make with each other. And if that shape is convex, we say it's a weak trade-off, right? Because we can jointly achieve really good outcomes for both things that we're interested in. Whereas when we have this more concave shape, we said there's really strong trade-offs, because we can't, if we've conditioned on achieving really high juvenile salmon population size, for example, we can't get a really high negative proportion resistant lice. So we're kind of trading off our two outcomes there. So looking at the results from my model, on the left, you can see that we have a win-win scenario, right? We have that convex shape indicating there's relatively weak trade-offs in this case between wild salmon population size and economic costs. And this is particularly true when we have really high treatment effectivities. So when you're using really good treatments, we tend to see larger wild salmon population sizes and lower economic costs. But these same strategies, unfortunately, lead to negative evolutionary outcomes. They lead to really high resistance. So right, the light green circles are inside our frontier on our right graph. So this is indicating that by choice of treatment threshold and treatment effectivity alone, we can't capture all three of these benefits. And we might want to think about additional management levers that we can layer on this so that we're able to achieve more of our goals. So the lever that I thought about next was this idea of gene flow. So at the start of this, I introduced this idea that maybe lice from wild salmon populations could come in and kind of swamp out any selection happening on the farm. And that's what we wanted to explore here. So I have two examples of increased gene flow, increasing the productivity of the wild salmon population and increasing the size of the wild fish population directly. Across our x-axis is our treatment threshold, and then I'll again plot points for different treatment effectivities. What you're going to see on these plots is a gray bar, which gives the range of resistance proportion for our base parameter case. And then the points are for our new simulations. So when these points are lower than the gray bar, that means that the increased gene flow is slowing resistance evolution, which is what we ideally would like to see. And so these are our results. We can see that particularly when we increase the productivity of wild salmon, this can slow resistance evolution, though doesn't stop it completely in many cases. So again, reinforcing this idea that adding management levers, layering them on top of each other, can help us achieve all of our different management goals across conservation, economics, and evolution. For my last vignette today, I want to talk a little bit about how hosts adapt to novel diseases. So disease has caused really high profile population declines in species that we all love, like sea stars, abalone, and frogs. But at the same time, this has also led to signals of selection in some populations. So Payasastar, adapting to some degree to wasting disease, abalone co-evolving with bacteriophages, allowing them to survive withering syndrome, and frogs acquiring adaptive alleles that allow them to survive despite infection. And this is true across a range of species, including rabbits and Tasmanian devils and bats, as well as a range of disease ecologies. So in cases where disease has density-dependent transmission, like Tasmanian devil facial tumor disease, or environmental transmission components like white nose syndrome in bats. It's also true across a range of host response potential. So in cases where we think that hosts die following infection, like sea star wasting disease, in cases where there's some degree of host recovery, and in cases where we have acquired immunity following infection. And the part that I think is really cool is layered on top of this, we also see multiple adaptive pathways at play. So for example, we see the evolution of mortality blocking alleles in bats and frogs adapting to novel diseases. So in these cases, these alleles allow individuals that are infected to survive the infection at much higher probabilities compared to individuals without those alleles. We see recovery augmenting alleles to some extent in Tasmanian devils responding to devil facial tumor disease. So these individuals can get infected, but they recover from infection much faster. And then finally, we see some evidence for transmission blocking alleles in our abalone populations that have co-evolved with these phages. And in this case, this prevents the individuals from getting infected in the first place. So it's kind of our classic resistance to disease that we might think of. And what I was really interested in was thinking about if there was a way to quantify the likelihood of evolutionary rescue and extinction across all of these different kind of dimensions of disease ecology and adaptation. So to answer this question, I created an individual-based model. So in my individual-based model, I'm simulating a bunch of little individuals, and I give them qualities like their genotype, which determines their disease robustness. So in this case, our WW individuals are wild type, and they have low robustness to disease in the cream colors. Whereas our resistant alleles with our RR genotypes are much more robust to disease across one of these adaptive pathways, and you see them in the darker colors. At each time step, I simulate disease dynamics. So an individual can become infected, it might recover if that's possible, or might die. And then after disease dynamics occur, I simulate reproduction. So each individual produces some number of gametes based on a genotype specific mean. Those gametes are randomly recombined to produce offspring. And then the total population of adults and offspring is limited by the carrying capacity. So if there's excess offspring, they're randomly killed. I do this across a lot of different simulations. And for each simulation, I'm tracking things about the population size. So what's the population size at each time step? What's the adaptive allele frequency across time? And then what's the infection prevalence across time? So what you see here is a bunch of different simulations, and I want to highlight 4 that show different trajectories following introduction of novel disease. So the first is extinction. We know that there are some populations that go extinct following introduction of novel disease, so this is expected. The second is what I call infection loss only. So in this case, the population starts to decline due to disease. But when the population starts to get really small, it's a closed population, there's density-dependent transmission maybe, so the disease burns itself out. It's not able to transmit to a new individual. So our infection actually completely goes away. And then once there's no infection, there's no stress, so our population recovers. And what's really important here is if you look at our middle panel, our adaptive allele frequency is pretty low throughout the whole simulation. So in this case, there's not evolution that's causing the recovery, it's rather a stochastic sort of infection loss that's leading to recovery. Next, we do see signals of classical evolutionary rescue. So our population declines, our adaptive allele sweeps, our population recovers. So we get a nice U-shaped demography, a nice sweeping of the adaptive allele. And in this case, the pathogen might actually coexist with the host. So we see persistent infection after the rescue event. And finally, the last trajectory that I've noticed is what I call transient rescue. So in this case, the population declines due to disease. The adaptive allele starts to sweep up in frequency, but in cases where the adaptive allele provides some sort of herd immunity or benefit to the population, that can actually lead to extirpation of disease. So this increasing frequency of the adaptive allele decreases our frequency of disease, eventually causing us to completely lose the disease. The population recovers, and then once the population is recovered, there's no longer a disease around, we actually get back selection against the adaptive allele because it might be costly, and so it's not worth keeping around in an environment where we don't have disease. So an individual-based model like the one that I made has a lot of different parameters in it. And we wanted to think about how do we understand which parameters are most important for determining which sort of outcome we're in. If we're getting a lot of extinction, a lot of infection loss, or evolutionary rescue. So what I did was I did a global sensitivity analysis to create this map from parameter space to outcomes. So I drew a bunch of different parameter combinations from our parameter space. I narrowed down to 19 uncorrelated parameters for this. I simulated out those parameters a bunch of times and tracked how many times the population ended up with extinction, infection loss, rescue, no risk, or something that was undescribed by the outcomes that I was tracking. And then I created a map from our parameter space to our different outcomes using a random forest. And what this map does is it says which parameters are most important for going from parameter space to the different categories. So in this case, our global sensitivity analysis shows that things like disease outcomes, so for example, does the host die following disease, can have acquired immunity, is really important in determining what sort of outcome we end up in, as is the number of disease generations and the adaptive pathway, among a couple of other key parameters. Today, I want to focus in on disease outcome and adaptation pathway and give you some sense of why these things are important and what sorts of outcomes they're helping to control. So in the case of disease outcome, my interpretation or intuition about this is it's really kind of determining what our extinction risk is. So when we have immunity from the disease, there's just much lower risk of extinction overall compared to when we have, for example, mortality as our only outcome from disease. We tend to have much higher risk of extinction. So that's my sense of why that parameter is so important and what's kind of going on under the hood there. Thinking about adaptation pathway, we can look at within kind of one of those compartment structures, how does different adaptive pathways, transmission blocking, mortality blocking, or recovery augmenting alleles change the likelihood of extinction? And my sense is that in this case, the adaptive pathway is kind of dialing up and down the likelihood of rescue. So it's just some minor changes in if rescue is more or less likely, generally with mortality blocking alleles being the most likely thing to increase your probability of rescue. One of the results that I thought was really fun from this model that I wanted to share was this kind of idea of there's a danger in evolving in a host pathogen system. So we can think about what happens if we have no evolution, which is going to be on the right, and then as we start to dial up the strength of our adaptive alleles benefit. So if we have complete benefit of the adaptive allele, someone gets sick and they just will not die from infection at all, compared to if they get sick and they maybe have a 50% chance of dying. Right? So as we start to dial up the strength of the adaptive allele, I was curious to think about how our probability of extinction on the top and probability of rescue on the bottom would change. And I thought about this both in the case where we have a costly adaptive allele, which will be our pink points, and a case where we had no allele cost in our green points. So looking to our kind of case where we have really strong benefits of the adaptive allele, we see something that's maybe a bit expected, which is that strong adaptation is increasing our risk of increasing our probability of rescue and decreasing our risk of extinction, even when the adaptive allele is costly. So this is good. It's expected, and it means that evolutionary rescue is possible, particularly when adaptive alleles provide complete benefit. However, if we start to look kind of closer to our partially protective adaptive alleles, we see a really interesting pattern, which is that our probability of evolutionary rescue and our probability of extinction are simultaneously increasing. And this is because this sort of weak adaptation first prevents the loss of infection. So the infection is sticking around longer, increasing extinction risk. And it's also spreading much slower. So the same kind of phenomenon, we're staying at a small population size for a much longer time, which increases our risk of extinction. And so I'm tentatively saying this could be a potential evolutionary trap associated with partially protective adaptive alleles. So I'll wrap up by just touching on some key takeaways from each of my 3 chapters. So in my first chapter, I showed that evolutionary rescue is occurring in wild systems, but only under moderate environmental change. And the longer term consequences and the community level consequences of evolutionary rescue are still a bit unclear in a lot of these populations. Second, I showed that layering management levers together helped us achieve multiple types of management goals across economic, conservation, and evolutionary goals. And third, I showed that post-adaptation can allow evolutionary rescue, but can also increase extinction risk when the adaptive allele provides only partial benefit. And across these 3 chapters, the other thing that you might have noticed is just how different evolutionary rescue can look compared to our kind of typical vision of evolutionary rescue, where we have that U-shaped demography and the sweeping of the adaptive allele. So evolutionary rescue is much more nuanced than that across these different systems and in nature. So I want to end with this question that I've gotten from people after presentations and from reviewers, which is, if we start to expand our bucket of what evolutionary rescue is, do we really need evolutionary rescue? We have great concepts like microevolution that can maybe inform conservation. And it's certainly true, microevolution can help us understand things like resistance evolution, eco-evo dynamics, and conservation. But I think what evolutionary rescue really adds to this picture is it helps us think about extinction and compare and contrast extinction versus adaptation. And particularly, it's a nice way to bring these things closer together. So we're talking about extinction and microevolution jointly. And particularly thinking about how management actions are shaping extinction, how they're shaping microevolution, and how those 2 things are playing off of each other to potentially amplify or dampen the sorts of management actions that we're doing. So with that, thank you so much for being here and listening. I'm happy to take any questions, and I have a huge list of people to thank, as everyone does when they finish up their PhD. Happy to take any questions if folks have them. Yeah, Fernanda.
Yeah, fantastic talk. Congratulations. Yeah, so I want to know more about the results of the second vignette you showed. I got a bit lost with the figures. Could you go back to them?
Yeah.
I would like to understand them.
This one?
Yes, exactly.
Okay. Yeah.
Especially the second one
Yeah.
on the right. That was hard.
Yeah, it's a little bit weird because it's obviously not like this clean monotonic shape, which I think is what would be nice, but unfortunately, topology is a bit ugly sometimes. So what's happening here is as we move on this figure from left to right, we're increasing our wild salmon population size, and we're also decreasing our treatment threshold. So what's happening over here is we're never treating, and that's why we have really good evolutionary outcomes, but then we end up with a lot of lice, so we have very few wild salmon. As we start to dial that up, we start to select more heavily for resistant lice, but we start to get better populations of wild salmon because now there's less parasites on the farm. And then if we go all the way over, what's happening in that upper corner is a little bit funky, but it's sometimes referred to as the high dose refuge effect. So essentially you've treated the parasites so much that there's effectively 0 parasites on the farm. And all of those parasites that are on the farm their genes are getting swamped out by gene flow from wild salmon. So when the parasites from the wild salmon, they're all like susceptible, they hop onto the farm fish and they swamp out any signal of selection that's happening on the farm.
Can you say that in a different way? It's hard for me.
Yeah,
So this is trying to manage the salmon in the farm, not in the wild.
yeah, so the resistance proportion is looking at the proportion of resistant lice on the farm.
Okay,
So this is like on the farm, are there a lot of resistant lice?
okay.
Yeah, and the reason why there's not a lot of resistant lice in that upper corner is because all the lice from the wild fish are swamping out the selection that's going on.
Okay, I guess I needed that
Yeah.
information for the way access.
Yeah.
Perfect, okay.
Totally. Yeah, sorry, can I just add one thing to Fernanda's point here? The other thing is obviously that point would be great to achieve, right? Like we definitely got huge wild salmon populations, no resistance. But that point over there is this line right here. And so we can start to get really negative economic outcomes if we're not careful there. Joe.
So I know in your review, you're only focusing on animal
Yeah.
populations. I was curious, just like, if in the ones you've come across, like in plants and microbe systems, especially like, I don't know if the folks in like biomed want to call it evolutionary rescue, but there is like this big, scary, like microbes evolved to antibiotics kind of thing, and a lot of rapid evolution of like invasive plants where we might want to like impede evolutionary rescue, or I guess that wouldn't quite be evolutionary rescue in that case, but I was just curious, did you see like, just looking around, like any, even though it wasn't your main focus, like any like general qualitative differences in what the nature of research in
Yeah.
evolutionary rescue is based on like system or anything like that?
Yeah, so we definitely really focused on animal systems and kind of ignored plants completely, as well as microbes. Hey, Julia. So first I would say there's a really great review paper by Helena Alexander and colleagues kind of comparing and contrasting the medical vision of antibiotic resistance, they don't call it evolutionary rescue, with evolutionary rescue, and trying to bring those worlds a little closer together. In terms of plants and evolutionary rescue, I think a lot of the literature tends to focus on agricultural systems, to my knowledge. And so there's a lot more analogies with the kind of impeding evolutionary rescue world. in that sense. Yeah, Elizabeth.
So.
You have a comment?
Vignette. You had a situation with bimodal outcomes where there was a higher probability of extinction and a higher probability of revolving resistance. And you referred to it as a trap,
Yeah.
but I don't know if that's quite the right word I'd use, because what I predict is happening is that the outcomes are more bimodal. Either it goes extinct or there's evolutionary rescue, because presumably once there's evolutionary rescue, it doesn't go extinct.
Yeah,
Is that correct, that it's bimodal?
you can think
that
about-
sometimes.
yeah, you
get-
can think about this point, like these points
Right.
are all from the same simulation. So
Right.
it's like from 1,000 simulations, 30% are going extinct and like 2% are having rescue.
Mhm.
I don't know that I would call it bimodal in the sense that it's like a discrete distribution, right? So it's like a discrete thing. It's not a,
So
I
the
don't know,
question
say- you
is
can say
the
more.
rescue causing the extinction? Or is it
Oh.
that either you get
It's that-
rescued or?
so I would say the
you go
evolution, the fact that you're having evolution could be leading to a higher extinction risk and leading to rescue. But it's not that the evolutionary rescue is leading to higher extinction, but the evolution is what's increasing your extinction risk. Right here, you have no evolution.
Maybe. We could talk about it more later.
Yeah.
I am not convinced that the evolution, well, first of all, if they're rescued, they don't go extinct. But secondly.
Yeah, for those individual simulations, they don't go extinct.
Right, so the simulations are leading to one of two outcomes. Well, or there's things in the middle, but the simulations are either leading to rescue or they're leading
Extinction
to
or
extinction.
infection loss, right? That's the kind of third one that they're not showing.
Okay,
The other thing, Andy has definitely written on evolutionary traps and can maybe weigh in on this as well. I feel like you raised your hand, and I don't know if that's what you were gonna say,
Yeah, I
but
wasn't planning to, though. I think this seems pretty intriguing that they would evolve, but not quite enough, and that's a different sort of use of the words evolutionary trap than
Yeah.
what people usually do, but I
Okay,
think it's
that's fair.
kind of a cool use of that term. I mean, I was gonna ask my usual question of plasticity and all this, which could be even in the
Yeah.
review.
Yeah,
Are people seeing, because that's obviously,
Yeah.
most of the animals maybe do some
no, are
plastic
you reviewer
response.
one? Right, so
Yeah,
we got asked about this a bit. We kind of touch on plasticity in the review and kind of like, how do you differentiate when there's like plasticity versus actual like gene change, and what does that mean? And how is
Yeah.
plasticity like potentially inhibiting rescue or facilitating it, right? Like, is it something that buys time for rescue, or is it something that like gets in the way of rescue?
or just means you don't have to because
Yeah,
plasticity
well,
is taking care of the problem.
To some
well enough,
extent,
which could be this scenario here, where it's like, takes care of it just barely enough, but not enough.
not
and-
quite enough, yeah.
Yeah, that would be.
Yeah, I mean,
intriguing.
I think that people have thought a bit about it in like the climate change context, right? That's really where I've seen a lot of that literature go, and I don't think we have a great resolution to that yet. But there's definitely some discussion of plasticity. There's some experimental work really trying to pin down the role of plasticity in rescue and answer this question of like, is it sufficient? Does it hinder rescue? Does it amplify rescue? So there's definitely some emergent work there that's been
I think really intriguing could be whether or not past evolutionary history shapes both their amount of plasticity that they'd likely be, what they would likely do, and their evolvability. And whether or not there's like a trade-off there that if your past evolutionary history favored using plasticity, you don't have much, the genetic variation or something like that. I haven't seen literature on that, but might just not be on top of that.
Yeah, Caroline, we're at 5, so if anyone needs to go, please feel free to head out. No, you can.
Okay.
Sorry, I didn't mean to
I was, like, this is coming from your third vignette. I was just curious. So throughout your talk, you're talking about like allele frequencies. I don't know much about this, but in my system, I'm learning a lot about polygenic variation and polygenic,
Totally.
that's what's
Yeah, yeah.
driving these differences.
Yeah.
So when you model, do you, like here, when you're talking about these genotypes, does polygenic variation, can that also contribute to
Yeah.
evolutionary rescue? And how do
Totally.
you go about that as a modeler?
Yeah. So in this case, most of the disease papers that I've seen suggest that it's like a major effect locus and then maybe a background of small alleles.
Yeah.
And so what we do is we use a paper from Landy from, the 80s or whenever he was writing all of his papers that everyone uses. And we say that there's a genotype mean, and then that's given by your large effect locus. And then there's some variation around that, which is given by all your small effect alleles. So we're drawing from a normal distribution around the mean phenotype from the genotype. You could go like full polygenic and like model multiple traits, and that would be totally fine. It would just eat into a lot of computation time.
Yeah.
And you could also go full, like it's a single trait, and not consider any sort of variation around the genotype or the phenotype. There's kind of a range there, and we've chosen something that we think is a reasonable middle ground.
Thank you.
Thank you everyone for coming.